so when I stand up in front of an audience and say I'm a UKAS assessor, I'm expecting tomatoes to come at least as a minimum. Okay, so please, I'm going to try and put white's the wrong colour. What I'm going to talk about are two of the things that I think there's been a lot of debate on when assessors have been pitching up to laboratories and trying to get them accredited to ISO 15189. Okay, so what I want to do is I'm going to review the requirements of ISO 15189. I'm going to try and tell you what I think the difference between verification and validation is, and I'm going to suggest some of the analytical data I would expect to see as an assessor go into a laboratory, and also to try and explain that there are benefits to us as providers of the laboratory service by having this verification and validation data, i.e. it's not just for UKAS assessments and delivering ISO 15189. Okay, few introductory notes. These are my views. I'm not employed by UKAS. I do it as part of my professional duties, and I thoroughly enjoy doing them, although sometimes it does get a little bit confrontational. We try not to make it that way, but sometimes clearly there are differences of opinion. And one of the things I would say is that if you think you'd like to be an assessor, just put your name forward, because it's clear we don't have enough assessors. And I think the one of the big benefits of the old CPA and now UKAS is that we still have people who work in real laboratories doing the part of the assessment process. I think it would be not good if we had a process that was completely disjointed from laboratory practitioners. So these are my overall views of what I've seen since I've started doing UKAS assessments. Okay, so it's longer ago than UKAS assessments that I started doing CPA assessments. And I don't know of any lab that would not have gone through some sort of process when they were introducing something new. Okay, so if you've changed your analyzer and you're going to introduce it, you're going to do some analytical work to verify that that is giving you what you expect it to give. My observations on the early assessments to ISO 15189 is that the verification reports were quite limited. So you'd go into a lab and you'd say, okay, I see you've got a new analyzer, you had it delivered, the analyzer delivered 12 months ago, did you do a verification, can you show me some data? One of the things assessors are likely to do is to say, show me, okay? Because we want to see evidence, because what we're looking for is evidence of compliance with the standard. So, show me the evidence that you've done a verification. I've had a variety of responses from a fantastically written up verification through to a filing cabinet, pulled out some printed off data from a Excel spreadsheet with 11 compared results for urea with no clinically abnormal results and said, look, they compare. Now, that is when you start saying, actually, I think you've got a finding here that you haven't properly done that, okay? That variation that I've seen in laboratories has changed from the early UKAS assessments I've done to the more recent ones I've done. And some of the data is very comprehensive. I think there's a general point that if you have, and I hope most people now have, an SOP that says, if we need to do a verification or validation, I've got an SOP that guides me as somebody working in the lab about how we do it in our laboratory. If you've got that, you find a lot of consistency. When you don't have that, you see some verifications being done very well and some that are not done so well. So if you've got a big laboratory service, you might find that one part of your service is doing it very comprehensively and another not so well. So some broad definitions. Verification, to my mind, and in the words of the standard, is really about Siemens have an assay for urea, okay? And they have done some validation work in their headquarters, and they have said and made claims that you find in a kit insert. you don't have to repeat all of that validation work, but you need to verify that in your hands it is performing in the same way that Siemens think it should be performing. Okay? So, for example, a Beckman analyse method on a Beckman analyzer used exactly as the supplier has instructed. Okay, that would be a verification. However, validation, and I'm going to give you a bit more detail on this in a moment, validation is where, okay, you've got a HPLC method for vitamins A and E, okay, and you prepare the standard yourself, you prepare all the mobile phases yourself, you've dialed in the settings yourself. How do you know you've optimised that properly? So that's a much more comprehensive piece of work. And again, in a little while, I'm going to talk about some of the things that they have in common, because there's some things that you want to see in both of those types of reports. So I'm going to go through verifications. Now, whenever people say, well, what should I be doing? The first thing I say to them is, go and look at what it says in the standard, okay? And whenever I find something and I think that doesn't look quite right, I'll often talk to the people I'm visiting and say, I think that doesn't quite meet the standard. Now, before I write up a finding, I will go to the standard and I'll see what it says, because I don't want to make up the standard because I'm thinking this is a good thing to do, this is a professional thing to do if you're going to introduce this method. It's about what it says in the standard, okay? So what it says is you've got to have objective evidence against specific requirements. And I'm going to take each of these in a little bit more detail. So specified requirements, you need to say what you're looking for, okay? So if your new serum urea method, the manufacturer is claiming that it's got a precision of 5% across the concentration range, you perhaps need to state, I'm going to meet the manufacturer's guidance, manufacturer's claims, and it's going to be less than 5%. So you say it, something like that. In your report, hopefully. The objective evidence is to then do that precision analysis, repeating samples in whatever format you think is appropriate, and therefore provide objective evidence that it's met that specified requirement. So when do you use a verification? Lots of situations. So I've just talked about when you're verifying a new method coming in. Okay, so a verification is, if you go to a lab, I should think the majority are in that situation. You've got an old method and you're introducing a new one, or you have retrospectively verified your method because UKAS is coming along and the expectations of the verification are slightly different to what perhaps you've done in the past. And you want to make sure you pass your ISO standard assessment and you do a retrospective verification. But you can also verify other things. So, for instance, I'm sure all of us have recently introduced AKI alerts, okay? But when you did that, you had done some calculations in some sort of data management system, either a standalone data manager or in your LIMS or somewhere, and ultimately that's going to get onto a patient report. And what you might have done, and I think I might ask this question if I visited a laboratory, okay, so you've introduced an AKI alert. Have you checked that that's actually working properly by taking a load of screen grabs at the different stages of the calculation to make sure that it's actually ending up in front of a clinician in the way you think it should? So if it's an AKI alert and you think it's AKI level 1 and your report ends up going to level 3, i.e. it's miscalculated it, that's as a bigger mistake as if you've reported the value incorrectly of the creatinine. So another time is when you introduce a completely new method. This is a bit trickier because you've got nothing as a comparator, but in those situations you might get samples you refer to another laboratory, again to get some sort of comparison. And when you produce patient documents, you might want to verify that what you've written makes sense when it's given to a user. So for instance, something very simple, collection of a 24-hour urine sample. I'm sure we all have guidance for that process, or collection of a stool sample. So if you think about the guidance that we're giving out to users, you might want to verify that what you've given out is going to work properly. So you might have a document that describes that. So there's a number of different uses of verification. You might, for example, verify that your new temperature monitoring system is working in the way that you expect it to. So there's all kinds of verification reports, not just methods. And if we, again, read what the standard says, verification is applicable to validated examination procedures, i.e. the manufacturer's done it. So key thing here is you can't just believe what the manufacturers state, but equally you can't assume that the way you've set it up is going to be exactly the way the manufacturers have set it up. So for instance, a very simple mistake that can happen, for example, is you've got an endpoint chemistry on your general chemistry system and it requires you to plug a calibration value in. Okay, if you put that in wrong, you're going to have a bias, aren't you? So you're verifying that what you've done in terms of putting everything together is working correctly. Again, from the standard, the laboratory shall obtain information from the manufacturer, the method developer, for confirming the performance characteristics of the procedure, i.e. the manufacturer will give you the performance it's expecting to see. And some of the method inserts that you see from manufacturers are very, very good. And they're key things for you to get your head around when you're doing your verification. Final extract from the standard at this point is that the laboratory shall document, which means you've got to do it, okay, the procedure used for the verification and record the results obtained. That's best achieved, I think, in a report. So the situation where somebody says, yes, I've done it, and here's the data, and here's a spreadsheet, without any commentary on it, actually doesn't help very much to show that you've documented things. So what am I looking for when I look at a verification report? I'm looking for what was the intention of the report, what's it verifying? Is it the temperature monitoring system that you're verifying? Is it a urea method that you're verifying? You need to say who did what when, okay? Because quite often I've seen verification reports that has nobody's name on it, it doesn't say when it was done. It could have been last week, it could have been 5 years ago. And key things that I think are important to put in are data on reproducibility, on accuracy and comparability. I would like then that data to be reviewed with a commentary on whether the data has met the intended purpose. So you said what you're going to do, and now you're going to present some data and say, yes, it has met the intended purpose that I've defined. Very important to say, if you look at method comparisons, no two methods agree, do they? Has anyone found any methods that agree totally? They're always different. So you've got to put that commentary on. Is it going to make a significance to the interpretation of that result? Do you need to change your reference interval? If you change your reference interval, what will that be based on? And then finally, you need some sort of sign-off, somebody with appropriate authority. And that could be your head of department, that could be a committee, your quality group, it could be your management group, but somebody needs to do it. And again, embedding that about the process that you go through within your verification SOP helps you get that consistent. So for method reproducibility, I ideally like to see a third-party QC material. I've often done findings on people not using third-party QC materials. If you read the standard, it's not insistent on it, okay? It said you should consider. However, you need to show that you've considered it. So if there's an assay where I know there's 10 manufacturers of IQC and I'm choosing not to use them for a tumour marker, for example, and you're using a manufacturer's self-claimed recommended IQC, I think that's a problem because you've not yet got an independent check on your day-to-day confidence when you're running your QC. Method reproducibility on IQC is generally better than it is on patient samples. So I think the complexity of matrix of patient samples makes them a little bit more ideal, but I accept that most people would use the IQC material. I think there needs to be some sort of assessment within and between batches, and I think when you're introducing a method, that's what you need to put in. Longer term, it's good to include intermediate precision. That's where you've taken into consideration that on your initial method introduction, you might only use 2 different lot numbers of reagent. You'll have, over a relatively short period of time, you may have only used 1 calibration and 1 calibration lot number. And of course, that's not the reality of the longer term and the performance of the method as it goes on in time. So I think the important thing is that at some time in the future, you update perhaps with an appendix to your report to say, okay, now I've looked at the intermediate precision, and whilst it was fantastic over a 2-week period when I put the analyzer in, now, over 6 months, I know that all these variable factors have contributed to a total imprecision, a total uncertainty of Y%, for example. And when you look at that method reproducibility data, you need to comment really, I think, on what the manufacturers have claimed. What I've observed, both in my own laboratory and in those that I've visited, is sometimes the precision claims of a manufacturer are over-egged. They'll tell you it's a fantastic precision, and when you've got your intermediate precision, it doesn't meet it. And in those cases, I think the benchmark to use, and we're talking about performance specifications as a question after the last talk, but I think the key thing here is to comment on why you think the precision is not as good as you expect it to be, and also whether it meets the desirable specifications that have been derived and on the Westguard website, which detail many assays and tell you what your desirable goals are for performance. Now, at this point, I'm going to maybe deviate a little bit from what I was planning to say, because we did have a question about performance targets. Performance targets or performance goals are what you're trying to aim for, but they're based on intra and individual biological variability in usually a healthy population. Okay, so it's a goal, that would be the first thing. And for most assays, in my experience, you meet what's expected because the biological variation is very high, and therefore you can get away with quite a ropey method sometimes. However, I'm going to ask for a show of hands here. So how many people in the audience measure sodium in plasma or serum? Pretty much everybody, okay. So does anyone know what the precision target on the Westguard site is for sodium?
0.1?
It's 0.6%, okay. Does anybody's laboratory analyzer deliver that, even on a single analyzer where you measure sodium? Has anyone got an analyzer that does that? Okay, so we have all failed to meet a performance goal that's now been set by Westguard on his site. In fact, it's not Westguard who cites it, he just reported them, isn't he? Yeah. But none of us are achieving the imprecision target that we're expecting to see. It's important to say it's a goal. Does it mean that all the sodiums we measure are completely useless and of no clinical value? I don't think that's the case, is it? I think there's lots of areas where we need to improve performance targets, and really what we're setting these goals here, this is something we're trying to aim towards. But if you're not achieving it, I think a commentary that you're not achieving it, but I have discussed this with clinical users and we do not think this is an issue. Sorry, that's a bit of a simplification of what you should be putting, I think, but something to that effect, that you notice that it's not meeting the performance targets. Or if there's no performance targets at all, commenting there are no performance targets, but we consider for these reasons that it's okay, and it's still of clinical value for the intended purpose, then I think that's really what some of the reports are now stating, which I think is a good thing to do. Okay, so method reproducibility might be presented something like this. Some mean data, so this is some creatinine and percent CV, so highly reproducible enzymatic creatinine method based on 20 replicates. Generally in the report, I would like to see the analyzer serial number, the reagent lot number, the cal lot number, and the time period over which you've done that 20 replicates. And what a lot of labs are doing, and I think this shows that you're thinking about uncertainty, is to expand that with a measurement uncertainty expanded with a kappa of 2 to double up the SD to get you a mu measurement uncertainty. So it shows you thinking about measurement uncertainty. Now, I'm not going to be talking about measurement and uncertainty today. One thing I will say is, in most cases, most labs I visit, this is the limit to which they take measurement uncertainty. However, I think measurement uncertainty is a much bigger topic than that, because that's just telling you the precision. So if you think about potassium, to my mind, the biggest measurement uncertainty is how long it's going to take the sample to get from the patient to your laboratory. So I think, again, it's about trying to control that those things that are contributing to uncertainty in an overall ethos in your laboratory about trying to get quality results. So when you've got your data, you need some comments to say whether it's met what the manufacturer has claimed, and is it meeting the requirements for the intended clinical purpose. So again, that's back to your, okay, so maybe we've not met the goals, maybe we've not met what the manufacturers have stated, but is it going to be useful clinically to help patient management? And again, the liaison sometimes with users is very important in that context. Method accuracy, or method trueness, we can debate a long time, and I don't want to today, on the difference between accuracy and trueness, but generally what labs do is they'll take some old EQA samples. What I would say is ideally use new aliquots. In the last 30 or so years since I've been doing method comparisons, I can remember many times where somebody's dug out the old EQA samples from the freezer that are 12 months old, they've been kept at -20, you run them and you say, well, they don't agree at all. And you end up going, oh, the method's not working. Get some fresh EQA if you can. All the EQA providers are generally pretty good at helping you get some fresh samples. Some suppliers' products are limited, and therefore they may charge you for them. But I think actually to get a good assessment and stop you running around in the wrong direction, it's worth getting fresh EQA samples. You compare to an appropriate target, okay? So if you're sodium, you're going to go for the all-laboratory trimmed mean. If you're looking at something like prolactin, you might want to compare to your method group. And you may want to present it either as a table or as a graph. So for instance, this is some data on AFP, and we've got two different architects that we've looked at, and we've assessed whether the results of each of these EQA samples have, how close are they to the method group mean in terms of the number of SDs. So are they fitting in the method group, okay? And you might want to plot it. So you can see here that we've got the number of SDs. So really we're looking for, because obviously the distribution should be between -2 and +2. Here's our old analyzer which had a method-dependent bias. This is amylase. Sorry, I should have pointed out. So amylase is you're comparing to the all-group mean. And the architects, the C16000s that we were analysing here, are clustered around the consensus value, with most of them falling within ±1. So that's I think acceptable performance. Next thing to talk about is method comparability. So you've got an old method, you're going to introduce a new method. So you will want to compare old with new, and this is what most of us are doing as part of our verification. And for uric acid, in this case, we had an old method on the Siemens Alvia. We introduced the 16,000. So for uric acid, it's not that difficult to get a good concentration range. Okay, so one of the things that, again, I sometimes report findings on. So I did a finding once where, okay, so somebody's introduced the new CA199 method. In fact, they're introducing it for the first time in their laboratory, and they've compared a Roche method with a Roche method down the road, another hospital. They've got 12 samples, okay, CA199, 11 of them were within the reference interval. One had a value of 1,350, something like that. So really, really elevated, and they drew a line, and within the reference interval, they agreed pretty well. The high value was 30% difference, and when I said, have you got any other values? They said, no, that's all we've got. To my mind, that hasn't proved the intended purpose, because obviously with CA199, you're often using it in the context of monitoring a patient, and you want to know what the serial values will be. So if you've got a 30% difference, even with the same method on the same platform, that to me was a problem. and they commented, this agrees well in the normal range, and therefore we're going to live with the method. That, to my mind, was not testing the method to its appropriateness. I did a finding on that, and they repeated lots of values from the top of the reference interval all the way through to 1,300. And when they did it properly, they found that there was actually pretty good agreement. And of course, I think what the problem was is they had diluted that very high sample, and they had used different dilution strategies for each of the sites that were actually doing it. So it's worth exploring that properly. So back to my method comparability, what I'd like to see is, and again, I'm not being prescriptive, I'm just telling you what I like to do in my own lab and what I'd like to see and why sometimes we might pick things up. So this has been compared, so again, with passing and bad block analysis. Okay, so one of the things that, again, I think is a problem, I don't think I've ever done a nonconformity against it, I've generally done a recommendation, is that they will take an Excel spreadsheet and they'll do a straightforward linear regression. That straightforward linear regression assumes there's no error on the method you're currently using and assumes all the error is in your new method. That can't be right. Both of them have error, and therefore you need to do some sort of cleverer regression analysis. I prefer passing and bad block, but there's also deeming regression, and both of them can pop out on a number of statistical packages you can buy very, very at reasonable prices, shall I say. But passing and bad block assumes error on both the measures, and therefore it's a much better way of looking at that data. The other thing I think is really important to understand method differences is to do a Bland-Altman plot. Okay, I noticed in Clinical Chemistry's last month there was a classic citation from the original Bland-Altman paper, and they make some comments on how many times. So I think the Bland-Altman paper has been cited in the BMJ more often than any other paper ever published. Okay, so these difference plots are quite commonly seen, aren't they? So we, I think, should be using them as standard ways of assessing data. What I would say is if you use some of the standard packages, sometimes the graphical display doesn't look particularly great. So it's not centered this, which I'm not too keen on, and it's given a percentage bias here of 2.3%, but actually expressed it in out of, you know, a numeric value on this axis. But you get the gist that here you've got a consistent negative difference. But when you comment on this, of course, as I said, no two methods ever agree with each other, but this one, 2% difference, and I think this is uric acid again. 2% difference is not going to cause any problems. You may need to refine your reference interval or make some considerations of that. And of course, the important thing to remember as well is that you shouldn't just run the patient comparisons on one day. If you just run, okay, I've run 50 samples today, on my old method I'm going to run 50 samples today, you haven't taken into consideration the normal variability that happens on a day-to-day basis, the things that you see when you look at your Levy-Jennings plot. So actually taking it over a number of days or weeks so that you get a true estimate of the true relationship between the two methods is important. And of course, one of the things to comment on is over a period of time, methods can drift by 2%, can't they? The same method on the same analyzer, new calibration lot numbers, sometimes you see these sorts of differences on a regular basis, I would say. So contextualizing that is important. PTH, another method, Bland-Altman plot, difference plot. This one, of course, you would expect to see, with an immunoassay, a greater degree of difference between one method and another from two different manufacturers. So again, putting that into the context of how the method's used and whether there's a different reference range would be what I would probably like to look at. So just summarising properly what I've already said, no two methods are the same. So when you see differences, it's a matter of trying to contextualise them in how that method is going to be used. Sometimes that means you need to review your reference ranges. If you review your reference ranges, make sure you've got an evidence base on which to make that decision about whether you're going to choose a reference interval of a particular type. Is it the manufacturers? One of the things that I think has been very valuable is that none of us are going to do a comprehensive reference interval check for children, okay? We don't have the resources to do that. So looking at published data, so many of you I'm sure are familiar with the Caliper study from Canada, where they've done a lot of reference interval work on children, and now with multiple different methods. And that's actually very helpful, I think, to try and look at your own data. When you've made your decision on your reference interval, make sure you document it in the verification. And whether, again, you can find this out a little bit from the manufacturers. Are there any limitations you need to be flagging up to people? Is there a funny interference with that method that you have now that you didn't have before? So does hemolysis affect this method more than your last one? Because if it does, you perhaps need to do something about it. So things like that are important, I think, to put in your commentary. And finally, just reiterating what I said, the important thing is to have somebody in authority, I think that's the sort of wording in the standard, to review the data and say whether it's okay. And say, yes, we're okay to use this, we're going to start using this method from this date, and a date and time on that is essential. So validations, I'm not going to go into too much detail on this, because I think fundamentally, first of all, the definition, not too dissimilar wording, objective evidence, specific intended use or application, and there's a couple of notes on this, but you might want to do validation, not just when you've got your clever HPLC method for vitamins A and E, okay, which you've optimised and done yourself, but in these situations, as are listed in the standard. So non-standard methods, I think that's when you've so I've always struggled with the difference between non-standard methods and laboratory designed or developed methods. So I think generally what we're saying, non-standard methods, that could be manufacturer X has produced a kit and you've put it on as a user-defined method, okay? So it should work. It's very similar to perhaps the same manufacturer of your reagent and equipment, but it's non-standard because it's not the standard method that comes with that kit, whereas the laboratory design method is more the vitamins A and E on your HPLC. This third one is interesting, and I've been looking at this a little bit. Standard methods used outside their intended scope. So if you look at a method insert from manufacturer, it'll say something like, this method can be used with serum, or lithium heparin plasma, or EDTA plasma, okay? And it doesn't say anything about CSF, okay? So if it doesn't say anything about CSF, I think you need to be doing a validation and calling it a validation. You might not do a huge amount of additional work because you may justify why you're not doing additional amount of work, but I think that's important to put in. Fluids, okay? So who runs pleural fluids through their main analyzers, okay? We do it, don't we? You will not find any manufacturer who will make any claims about fluids. Okay, so I think in that situation you needed to do some validation work, okay? Now I think pleural fluids behave very, very similarly to serum or plasma, but providing evidence of that is actually something I think if you want to get it in your scope, you're going to have to do. And again, what does the standard say? So the standard, and I've split this up into a bit that I've put in italics and bits that are not italics. The bit in italics are the bits that I've just gone through that you do for verification. They're exactly the same. You may want to do it a bit more comprehensively because you have a little bit more anxiety, or you want to prove in a little bit more detail. But you may also want to do the things that hopefully, if it was a validated method by a manufacturer, they would include. So specificity, are there interference substances? So for instance, if you've got a cortisol assay that you're running by mass spec, okay, you set it up and you think, okay, that mass transition is going to be associated with cortisol, because that's what it says in the literature, and we look for it and it's there and it seems to correlate. But what if you've got other similarly structured glucocorticoids or mineral corticoids in your sample that could potentially have similar transitions? You may want to explore those, okay? But measuring interval, what's your lower limit, what's your upper limit, what's your detection limit, what's the quantitation limit, and potentially things about diagnostic specificity and sensitivity. Okay, so that's from the standard. So I'm not going to say much more on validation, but other than say you will need to put a little bit more data into there, dependent on the type of method it is, okay? But this slide, I think, is trying to explain why I think it's important for reasons other than convincing a UKAS assessor you've done the right thing and you're going to get your accreditation. So I think it's really good for training, okay? In our lab, we get every member of staff who's new to the department to do a verification. And I think that's important because it takes them through the delving into a method and the basics. I think these are basics of how methods work. I think it's quite important. So for their registration portfolio, we have that as part of their evidence. And of course it helps you as well because you delegated work, aren't you? I like to delegate work, and that's why I had a whole day here. I've delegated work at the lab. But I often get emails like this, and when I prepared this talk at the weekend, I had two questions in my email inbox, and one was the hepatologist, and he said, or she said, sorry, in 1997, I'm looking at some longitudinal monitoring of some patients with some liver condition. I won't bore you with the details, but you had a reference range in 1997. Where did it come from? And if I asked anybody here, in fact, probably a lot of people here weren't even working in 1997, but if I asked you about your laboratories and what reference range you were using in 1997, I would hope most of you will be able to give me what it is, because you can probably dig that out of your limb system. But the justification of how you came to choose that reference interval may be lacking. So, as I say lacking, you might be able to find it, but it's going to take you about an hour, 2 hours a week to get that data. And that's the difficulty I've got, because I know it's somewhere, and I've got to find it. Now, we upload all our verification reports onto our iPassport system, our document control system, and therefore, we can just plug on it and say, okay, we're using this reference range at the moment, and we based it on this decision. Okay? Also, a paediatrician asked us to pull some data on patients with congenital hypothyroidism, and it dates back to 1999. Okay? And during that time, you will have seen several method changes, and we saw 3 method changes during that period of time, and each of these, when you transition to the new method, there's going to be a difference. So if you're looking at longitudinal monitoring, how much do you take that into consideration when you're looking at that data? Okay, and I think if you've got your verification reports available, you'll be able to pull that out. So, that's the end of my talk that was about the UKAS process, but I just thought I'd share with you, because it's a Digestive Diseases Day, I'm just going to talk about fit testing, because we have started running fit January this year, okay? And this is some of the data, not all of it, that we put into our verification report. Okay, so reproducibility, okay? Here, sampling is important. We've heard about that all day, okay? So what's the variability at different concentrations? So if you're just running a sample in a single dipped stool sample, you've got very good reproducibility, 2.9% at a concentration of 35. But what about these samples? These are repeatedly dipped in the same stool. Okay, so you've got quite large CVs. So the greatest variability is associated with the sample process. So we have some commentary saying how important that is in our SOP. We have some data in there in our verification report. And it deteriorates quite markedly. So this is at level 2. So interestingly, I've written on the board what's our cutoff. We're using 7, and I won't bore you with the details, but we decided 7 because that's the lowest calibrin value. And also, if you look at the precision profile, it's about 20% when you draw the line through the data on the precision profile. Somewhere between 16 and 2, you get greater than 20% in precision. But comment on it. EQA, we ran some EQA samples. There is a pilot EQA that Jane French is running, and we ran those samples. The problem with this data, from my mind, is there were only small groups of, I think there was only about less than 10 people contributing to this at that point in time. And whilst I was assured that we were getting the overall consensus right, what I was more concerned about is this is the amount of micrograms of hemoglobin spiked into the stool, and this is what we recovered in terms of the value. So I think there's quite a lot of work to be done yet on EQA, on calibration for FIT. And of course, you do a calibration, a comparison. So we had not stopped doing fecal occult blood. Interestingly, if you look at the figures on it, I think 46% of laboratories had stopped completely doing fecal occult bloods. There's a paper on that from a couple of years ago in terms of what's available to GPs in terms of direct access. But you can see that when we did a FOB-FIT comparison, we found quite a lot of differences with 77% agreement, and noticeably a drop in the number of FOB, the positive fecal occult bloods with the GWAC method compared to the FIT method. And of course you think, well, the old method is so rubbish, I'm not surprised by that. And therefore it must be this new method being the right result, because it is better, isn't it? It's an analyzer, it's got to be better. But that inherent mental bias, you need to back up with some evidence. Okay, so what we did is we said, okay, let's follow all of these patients through to see whether they've had endoscopy and histology that helps us clarify which one is the one that meets the specific requirement. Now, after all this morning's discussion, saying that this is for the detection of cancer may be a bit strong, but basically that's what you're trying to do, isn't it? But if you get all the histology data out, what we found was that it had equivalent sensitivity for picking up adenocarcinoma patients, but a much, much reduced number of false positives. And that's probably because it's less, well, it's not affected, as far as I'm aware, by all these dietary things that we've recommended patients avoid, like avocado, or whatever it is, raw meat, all the things that we advise patients not to take if they're doing collection of stool sample. So I think what we observed was a very big reduction in false positives. And we captured that data in our verification report. Okay, so come to the end now. So I think verification and validation reports are really valuable. Okay, they not only get you through your ISO 15189, they'll help you run your laboratory a little bit more favourably. I think the key things I'd advise is read what the standard states. That's not just for verification and validation. Look what it says about anything. So if you're uncertain and you're having a debate at work and you're having an argument because somebody says, I don't think I should be doing that, and you say, oh yes you should, oh no you shan't, I think the important thing is to see what the standard says. So if the standard states something, then that's what we really need to try and go on. It's a dry old document. Has anyone read it in one night? It's quite good going. But I think the reports can improve the way our labs work. Okay. Thanks for listening.